Hi, thank you. Yes, so my name is Heather Veilleur. I've got the challenging French-Canadian last name. And I'm going to talk to you about some work I've done this year with my colleagues at the Center and JCU in collaboration with some scientists from King Abdullah University of Science and Technology in Saudi Arabia. So we all know that the oceans are getting warmer and warmer due to climate change. The IPCC predicts that tropical regions will warm by as much as 1.5 degrees in the next 50 years and 3 degrees in the next 100. Already we're seeing the negative impacts on marine organisms with dire predictions for the function and productivity of marine ecosystems in the future. Tropical species may be especially vulnerable to these increases in temperature. Because they've evolved in relatively stable thermal environments, they may have lost the ability to cope with the changes in temperature predicted over the next century. In this model of species fitness, you can see that the tropical species in the top panel has a much narrower thermal range compared to that of the more temperate species below it. As such, it's already living near the extremes of its thermal tolerance. At the predicted temperatures by the end of the century, the tropical species will likely suffer major declines in fitness as it experiences temperatures beyond the maximum of its thermal range. In an attempt to discern how marine fishes might be able to cope with increased temperatures, many studies have exposed them short-term to increased temperatures for hours, weeks, or months. And we've seen exactly what's predicted, declines in performance and sometimes even death. For example, in 6 equatorial reef fishes were exposed to current and elevated temperatures for 14 days. Half of the fish suffered declines in aerobic performance at +2 degrees above current day temperatures, and all fish suffered declines when exposed to +3 degrees, with one species unable to survive at that temperature. Similarly, in the Spiny-chromis damselfish from the Palm Island region on the Great Barrier Reef, adults were exposed to high temperatures for 7 months. And they showed decreases in reproduction, development, and growth, suggesting an overall decline of this population in the future. But climate change isn't going to cause the oceans to rise in temperature that quickly. It'll likely happen over the course of generations. So how will these fish cope in the longer term? Well, there are 3 main survival strategies. The first is to find a cooler, more suitable habitat, either by migrating to a new reef or finding a cooler spot on their home reef. But not all species are going to be capable of doing this. That leaves adaptation and acclimation. Adapting is survival of the fittest, so those that already have favorable genotypes in warmer waters will survive and pass that information on to the next generation. Then acclimation is moving away from the genotype, and it's being able to alter your phenotype, so adjusting physiological, behavioral, or morphological characteristics to better suit an environment. This is thought to occur by an underlying mechanism, so by changes in gene expression or energy allocation in order to maintain an optimal phenotype in suboptimal conditions. Think of it sort of like an underlying buffering mechanism. So with increasing temperatures, marine organisms may have a reduced capacity to take up oxygen and deliver it to organs and tissues. Resting metabolic rate is the amount of oxygen required to perform vital body functions at rest. Maximum metabolic rate is the maximum amount of oxygen that can be taken up. And aerobic scope is the difference between the two and represents the capacity for performing aerobic functions such as swimming, foraging, and defense. Now, with increased temperatures, there might be an increase in the amount of oxygen required to sustain the animal at rest and a decrease in the maximum amount of oxygen that can be taken up. This decreases the overall aerobic scope and thus decreases the ability to perform those aerobic functions. But through acclimation, we might see a return of these levels to optimal levels. In a longer-term look at that Spiny-chromis damselfish, we've got the experimental design in the top panel. And there are 3 temperature treatments. So in blue, we've got the control treatment. Orange is an increase in 1.5 degrees and red in 3 degrees. And if you look at the bottom 5 fish, these are the 5 treatments that we're using. We've got control, developmental fish that were exposed from just after hatching throughout life, and then we've got transgenerational treatments that were exposed for 2 generations. And when we look at aerobic scope, we see that the transgenerational fish were able to fully acclimate compared to the developmental fish. And this was really interesting because it shows that there is this capacity for transgenerational acclimation, and it also highlights that short-term studies might be highly misleading for the long-term prospects of these marine animals. So now that we know that this process is possible, we need to find out how it's possible. If we can identify the molecular mechanisms of transgenerational acclimation in the Spiny-chromis damselfish, it might not only show us how this remarkable process is happening, but also give us a broader understanding of the trade-offs and limitations cellularly beyond just looking at the phenotype. It can also hopefully lead us to determine if the mechanism is universal across other species and potentially identify the species that are most at risk. So that brings me to the research that my colleagues and I have been doing, and we were identifying the molecular mechanisms in the Spiny-chromis damselfish, so particularly gene expression, so those developmentally and transgenerationally exposed fish. So in every cell of an organism is the entire complement of DNA, all of the genes. And when a particular gene is needed, a copy is made into RNA, and then the RNA is translated into protein, which are the structural and functional units of all of our cells, tissues, and organs. But we don't need the same proteins or genes in every single tissue at every single time point. And so, for example, if we look at the liver and the brain, you don't need the same genes in the liver and the brain or in the same quantities. You also, if you get a diseased liver, you might need to increase certain genes or decrease other ones. So if we take a snapshot of the RNA, so the gene expression in those transgenerationally acclimated fish, we might get a good idea of what genes and cellular processes are required to acclimate. So in collaboration with Kaus, we sequenced, assembled, annotated all of the RNA from those 5 treatments. We identified over 89,000 sequences, of which 87 were differentially expressed among those 5 treatments. To further identify which ones of these genes might have a direct role in acclimation, we correlated the gene expression to the metabolic data for each individual fish and found that 53 genes were significantly correlated. This is a heat map, and it shows the gene expression for each of those 53 genes that are listed at the bottom for the 4 treatments relative to control. So we've got the developmental treatments at 1.5 and 3 degrees in the top 2 rows, and the transgenerational treatments on the bottom 2 rows. So the rectangles that are in red are those that have higher expression relative to control, and in blue, those are lower expression compared to control. So the first thing to note really is that there are far more genes that are higher in expression compared to control, and that the transgenerational treatments have more highly increased genes compared to the developmental treatments. Now, if we take a look at just that large group here, they're separated based on expression patterns. We see that the majority of the genes are correlated to that acclimating trait aerobic scope. And they're involved in several different processes such as metabolism, stress and homeostasis, cellular transport and organization, and immune responses. And today I'm going to talk about the metabolism, which can be further broken down into lipid, protein, and carbohydrate metabolic processes. If we take a look just at the first 20 most highly changed genes in the transgenerational fish, 15 of them are involved in metabolic processes. The increases in lipid metabolism might be to maintain the proper fluidity of cellular membranes. So in warmer and warmer conditions, fish need to change the fluidity of the cellular membranes, making it more viscous. So they might have to increase the number of saturated lipids in their membrane. And the increases in genes involved in lipid mobilization and carbohydrate and protein digestion likely serve to increase the energy production in those transgenerationally acclimated fish, thus to improve aerobic scope. One of the really interesting genes was apolipoprotein E beta, and it had significantly higher expression in both the developmental and transgenerational treatments, and it was correlated to aerobic scope, and has major roles in triglyceride and cholesterol homeostasis, with potentially many other roles such as cellular plasticity, regeneration, and survival. It's also, along with other apolipoproteins, has been increasing expression in short-term fish studies. So it likely has a role not only in short-term thermal stress, but also in long-term acclimation of aerobic capacity. So a typical molecular tool to evaluate how fish and other organisms are going to cope with short-term thermal stress has been to evaluate the expression of heat shock proteins. And some are even inferring long-term capacity to deal with higher temperatures based on the ability to increase expression of these genes. So when a cell undergoes thermal stress, proteins tend to unfold and clump together and really decline in their normal function. So heat shock proteins are expressed in order to help them refold and return to normal. And when we took a look at all of the heat shock proteins and all of the RNA of all the treatments, and there were quite a lot, not a single one was significantly different in either the developmental or transgenerational treatments relative to control. And this really suggests that heat shock proteins might not be good indicators for the long-term capacity of adjusting to climate change, at least for the Spinochromis damselfish. So in conclusion, we identified the 53 key genes and cellular processes involved in transgenerational acclimation. This was for multiple different cellular processes, in particular metabolism, to likely improve membrane viscosity and increase transgenerational energy levels, and also found that heat shock proteins might not be appropriate long-term markers for acclimation. And this was really just the start of a lot of really interesting molecular projects. So now we're getting into the details of exactly how these genes were able to change so quickly after just 2 generations. And we think it's a process called epigenetics. And in acclimating aerobic scope, there were some trade-offs in reproduction, so I'm starting to look at gene expression in the brain and the gonads to look at what's happening there. Also looking at some 2 populations of the Spinochromis at different latitudes with different abilities to acclimate, and looking at what genes might be driving this difference. And in collaboration with Dr. Jody Rummer, we're starting to look at some of those genes that I identified, like ApoE-B, in some other fish exposed short-term studies, potentially as a predictive molecular tool for how species might be able to cope with higher temperatures in the longer term. Thank you.